Lesson 7, Chapter Cell, the Unit of Life. So in this video, now I'm going to discuss about the cell organelle, which is called as Golgi apparatus, or it is also called as the Golgi complex. This is also another membranous system like that of the endoplasmic reticulum, and the structure that is also quite similar to that of the endoplasmic reticulum. The discovery of this structure, it was in the year 1898 when Italian histologist and a Nobel laureate Camillo Golgi, while investigating the nervous system, that is the nerve cells of owls and cats, that he discovered a structure which is like that of a reticulate system or in a reticular form, and that is how this subcellular interreticular system, it was called as the Golgi apparatus or the Golgi complex. That is after the name of the scientist who discovered it for the first time. But the electron microscopic structure, it was described in the year 1954 by two scientists named Dalton and Felix. Now let's come to the location of this Golgi apparatus. The Golgi apparatus, they are mostly present in all the eukaryotic cells, and especially in case of those cells which are secretory in functions. That means they secrete, say for example, chemical substances like hormones, then enzymes, or such other secretions that is done. Such cells, they have more of this Golgi apparatus, and it is also found to be absent in case of some eukaryotic cells, such as it is absent in the mammalian RBC cells or RBCs, or it is absent in case of the male gametes in bryophytes, pteridophytes, and of course it is totally absent in case of prokaryotic cells. And for the other eukaryotic cells, so somewhere they might be less in number, or in case of some they might be more in number, and also its position in the cytoplasm that may change according to the type of cell. Say for example, in case of yeast cell, the Golgi apparatus that is found scattered in the cytoplasm, but in case of a mammalian cell, so it is found near the nucleus, or it is right beside that of the endoplasmic reticulum. Now, in case of invertebrates and the plant cells, usually they have some small Golgi complexes, and that is several in number, numerous in number. So these Golgi complexes, they are not one system, but they are scattered throughout the cytoplasm as small Golgi complexes, and such complexes in case of the invertebrate cell or the plant cell, it is called as dictyosomes. So there the term dictyosome is applied. Now let's discuss the structure of the subcellular structure. I am calling this to be a subcellular structure because this is a part of the cytoplasm and they are separate bodies there in the cytoplasm. Therefore, we can call them to be the subcellular structure as they are individual entities within the cytoplasm. So in the structure of the Golgi apparatus, here also it is quite similar to that of the endoplasmic reticulum. So generally the structures, they are made up of three parts, or they are found in three forms, that is the cisternes, then comes the peripheral tubules and the vesicles. So we can call the Golgi apparatus to be a centrally stacked, parallel, flattened, intercommunicating sacs or cisternes, and it is supported by the peripheral tubules and the vesicles, which are scattered in the cytoplasm of the cell. And they may vary in their shapes and size according to the type of cells. Now let's come to the first part of the structure of Golgi apparatus. As you can see in the diagram, the major part of this structure that is covered by the cisternae. So it consists of a stack of generally 4 to 8 membrane bound saccules or cisternae, and these cisternae, they are smooth but of variable thickness. So these cisternae, they are similar to that of the endoplasmic reticulum, where they are found in sac-like forms, which is filled up by matrix towards the inner side, and they are arranged parallelly with each other. There, the cisternae, it have two ends. One end, that is the convex face, which is called as the cis phase of the apparatus, and the other end, that is the concave end, and that is called as the maturing phase. So we can find two phases of this cisternae, that is the cis phase, which is found to be in convex form, and the maturing phase, which is found in concave form. And they also have almost similar function like that of the cisternae of the endoplasmic reticulum. Then comes the tubules. So tubules, they forms a complicated network towards the periphery and the maturing phase of the apparatus. That means the tubules, they are found towards the concave end of the cisternae, or they are connected towards the concave end. And as they are in tubular form, so they produces an extensive network of itself, and they are always lying towards the periphery, that is towards the side of the cisternae. The diameter of these structures, it is around 30 to 50 nanometer, and they are connected with the cisternaes. Then comes the vesicles. So vesicles, they are small, oval, round sacs of 20 to 80 nanometer in diameter, and they are generally found attached to the tip of the tubules. And it is of two types. One is the smooth-walled and another one is the coated. The smooth vesicles, they contain secretory substances. That means they are secretory in function, and so they are also called as secretion vesicles. Also, there is another part which is called as the Golgian vacuoles. So they are expanded part of the cisternae which have modified to form the vacuoles. So it is found on some kind of cells. So this is all about the structure of the Golgi apparatus, which is quite similar to that of the endoplasmic reticulum. Now let's discuss about the functions of this Golgi apparatus. The first function of the Golgi apparatus is that it helps in the packaging of materials that comes from the endoplasmic reticulum, or that is synthesized in the Golgi apparatus itself. So it acts as the packaging unit of a cell, where it packages or stores or modifies it. We can say that to be the modification. It helps in the modification of materials that comes from the endoplasmic reticulum, or the one that is synthesized in the Golgi apparatus itself. Then comes another function, which is the synthesis of carbohydrates. So this Golgi apparatus synthesizes some simple carbohydrates such as the galactose and certain polysaccharides such as cellulose, hemicellulose, pectin, etc. So that is their synthetic function. Then the third function, that is formation of glycoproteins. It helps the carbohydrates to bond with proteins. As I have said, that it helps in modification from the materials that comes from the endoplasmic reticulum. So the one which is formed there in the endoplasmic reticulum or in the Golgi apparatus itself, so here it gets modified, and hence a particular molecule called as glycoprotein. Now what is glycoprotein? So that is the carbohydrate when it forms a molecule with the proteins, that is called as the glycoprotein. So that is formed there in the Golgi apparatus. Then similarly, formation of lipoproteins. That means lipids and proteins coming from the endoplasmic reticulum are complexed into lipoproteins in the Golgi apparatus. Then another important function, that is the formation of lysosome. Lysosome, that is another cell organelle which have a very significant function in the cell. There the Golgi complex gives rise to the primary lysosome by budding from the trans phase of cisternae. Now the maturing phase, that is called as the trans phase, or the concave end of the cisternae, that is called as the trans phase. So the lysosome, which is another cell organelle, it is formed from the Golgi apparatus, and these lysosomes, they are formed by the budding of the cisternae which are present in the trans phase. Then comes another very important function, that is the formation of acrosome. Acrosome, that is a part of the male sperm, human sperm, or the human male gamete. As you can see in the diagram, that this is a structure which is found right at the tip of the sperm cell, that is a male gamete in case of human beings, and that particular cap-like portion, that is called as the acrosome. And this acrosome, that is given rise, or that arises from the Golgi complex. Hence the Golgi complex gives rise to the acrosome in an animal sperm. So these are few functions of the Golgi apparatus. So let me repeat it again, that is the packaging of material, then synthesis of carbohydrates, then comes the formation of glycoproteins, formation of lipoproteins, formation of lysosome, and formation of acrosome. That becomes this becomes some of the important functions of the Golgi apparatus. So in this video, I have discussed about the structure and the function of the Golgi apparatus along with its presence and absence in the types of cell. Thank you.